Well, hello everyone, and welcome to today's webinar, which is entitled Tar Spot Trends and Management. I'm Chris Boomsma, Director of Education here at the American Society of Agronomy, and it's my privilege to be today's moderator and to introduce the speakers and handle the question and answer period at the end. So with that, we're going to get started right away because we've got a lot of great speakers with us today that I want to get through introducing. But before I get to that part of the program, first, our sponsorship. This webinar series is generously supported by the Crop Protection Network. You may also have heard of them as CPN. The Crop Protection Network is committed to creating and enabling collaborative scientific outputs addressing the principal issues impacting field crops today. CPN's diverse team supports the mission of extension and land-grant universities throughout the United States and Canada, providing unbiased and accessible agricultural information for all. So thank you very much, Crop Protection Network, not only for the sponsorship of this series, but also for bringing us so much great content in the area of pest management, which is an area traditionally we struggle with giving out a large amount of CEUs in. It's hard to find great content, so I really do appreciate what we have here today with all of you joining us. All right, moving on now. A few housekeeping items before we get started. As always, if you have issues, registration, technical issues, this is very important. Please reach out to Lacey Edwardson first. You can see her email address and phone number on your screen at this time. If you cannot see my screen that I'm projecting to you right now, the number you can reach her at is 608-268-4953. Again, 608-268-4953. Note that this webinar is worth 1 CEU in pest management or professional meetings based on your certification. We will have a question and answer period at the end of this event. It'll probably be about 5 to 10 minutes in length. So submit your questions to me at any time in the questions box, and I will pose them to our presenters. Note that this session is being recorded. Both the recording and the slides will be made available to you about 1 week after completion, but often sooner than that. Please be on the lookout for future webinars by the American Society of Agronomy in the coming months. It's going to be a very heavy webinar season, November, December, January, February, March, so be on the lookout for that. Importantly, this webinar is part of a larger 8-part series. 3 of these webinars are now posted on our website. Take a look at those, they're coming up, and we will continue this series into 2022. So you have an opportunity here to learn a lot in disease and pest management. So please be on the lookout for these webinars and register for them, because they're going to cover a whole series of things. All right, now moving on to introduce our speakers. And as I introduce them, one by one, they're going to come on with video today. And it's going to be a pretty interactive session between our host of speakers. And so they're going to come on and share their visual with you too. So our first one is Martin Chilvers, who's an associate professor at Michigan State University. In this role, he leads a field crops pathology program encompassing both basic and applied research and extension. This program currently emphasizes seedling and root rot diseases of field crops. Soybean, sudden death syndrome, white mold, and tar spotted corn. He earned his bachelor's in ag science and his PhD degree in plant pathology from the University of Tasmania. Welcome, Martin. Thank you for joining with us today. Next up in our program is Darcy Telenko, who's an assistant professor and extension field crop pathologist at Purdue University. In this role, she has statewide responsibility for plant path research, extension, and agronomic crops in Indiana. She principally works at understanding soil-borne and foliar diseases and their impact on Indiana agriculture. Prior to this role, she was an extension vegetable specialist at Cornell, a postdoc at the University of Florida, and a research scientist at Virginia Tech. She has a bachelor's in biological sciences from Cornell, a master's in plant and soil sciences from SIU or Southern Illinois University, and a PhD degree in plant path and crop science from North Carolina State. So Darcy, thank you for being with us today. Moving to our third of 4 speakers. Our third is Albert Tenuta, who's a field crop extension plant pathologist for the Ontario Ministry of Agriculture, Food and Rural Affairs. He's held this position since 1991. Albert's involved in many North American regional disease management research projects and extension initiatives, such as is today, the Crop Protection Network, the SCN Coalition, Corn Disease Workers, and the Tar Spot Working Group. He has a bachelor's in plant pathology from the University of Toronto. Albert, welcome. And now our 4th speaker, Roger Schmidt. Roger's a software engineer at the University of Wisconsin-Madison. During his career, he's created 12 successful farming apps, currently in the Apple and Android stores. Brings a broad understanding of emerging digital technologies to the ag community. He's co-founder of Field Profit Inc., which is a recent startup focused on creating easy-to-use disease management software for ag professionals. He has a bachelor's in mass communication from University of Wisconsin-La Crosse. All right, very good. So with that, we've done all our introductions. Here they all are. And with that, Martin, I'm going to hand it off to you now, sir. And when you're ready, you just go right on ahead and show your slides. We look forward to everybody's presentation today. Thanks for being with us.
All right, thank you, Chris, for the introduction. Okay, so let's get started. So we're going to talk about TaskSpot obviously today, and I want to back up just a little bit because we probably have people here that are certainly quite experienced with TaskSpot, but we may very well have those that are not dealt with especially the full force of TaskSpot. And we sort of want to emphasize, that it can be a very serious disease. Here are some sort of typical symptoms we may see in a field, and we're going to go through about how to scout for that. So TaskSpot's been in the US since 2015. It first arrived or was first found in northern Illinois and Indiana. And it was pretty quiet for a few years. We're starting to find it in a few different places, including Michigan and Wisconsin. But it wasn't until 2018 in that really wet summer that we had an explosion, an epidemic of the disease. And that was really centered around southern Lake Michigan. And we'll get into a little bit about the spread of this disease here. And these couple of photos near Lake Michigan really sort of emphasize how quickly this disease can develop and potentially how devastating it can be. So in this particular field, the farmer first noticed symptoms July 8th. He'd had the disease previously in 2016 and seen it. He rotated to soybean, came back to corn. But because we had those wet conditions in 2018, lots of frequent rainfall, we had very serious disease develop. And you can see that on the left-hand side here. We had disease reaching up and even forming structures on the tassels of some of these plants. And so you can see those browned out areas. That's all TaskSpot that's moved up in the canopy and starting to shut those plants down. On the right-hand side here, 2 weeks after that first shot, the plants prematurely senesced. So September 7th that year, they were done. And the other important piece, as well as that obvious sort of yield reduction, there's very significant lodging potential here. So these heavily infested areas correspond to this area of the field that lodged. So that's just something else to be really aware of with this particular disease, especially if it's new to your particular area of the country. In terms of scouting for a TaskSpot then, it is relatively easy to identify. I would say it's harder to scout for the northern leaf blight and grey leaf spot just because those initial lesions are very small and are pretty easy to overlook. I think we've all certainly been out with growers and it's easy just to overlook things. So what we're looking for there is these raised black structures on the corn leaf, and they will be protruding through to the bottom side. So turn that leaf over and see if it's pushing through onto the bottom side. It can basically form on any leaf tissue, so husks, leaves, we very rarely sort of see it on stems. So it's really on that leaf tissue and sometimes on tassels. One of the other types of symptoms we see is what's called a fisheye symptom. And so we get some necrotic tissue, dead tissue around that TaskSpot structure itself. So that's just something else that we see sometimes, and it varies field by field. In terms of what it can be confused with, unfortunately, we have had some fields that have been sprayed because of bug poop. It didn't turn out to be TaskSpot, so it is really important. They certainly do look similar at first appearances. Bug poop, of course, on the left here, that can be just rubbed off or washed off the leaf. So that's pretty easy, and the bug poop isn't going to be protruding through to the bottom side, right? Obviously, it's just going to be on whichever side of the leaf that bug pooped on. And then the other one that's probably slightly trickier is southern rust or common rust teleospores. So once the initial infections are rust, that orangish yellow color, but once they form those teleospores, they tend to be like a blackish color, a little bit more like TaskSpot. So there, if you look at them closely, the leaf epidermis has more of a burst appearance where those structures are bursting through the leaf. So that's how we differentiate it. If you have any questions about that, make sure you contact your extension specialist and get a sample into the diagnostic clinic just to know what you're actually dealing with. That's very important. So what causes TaskSpot? Well, Phylochoromatus is the fungal organism that causes this. And it was described in Latin America back in 1904. So it's been around for a while, but only, as I said, recently introduced to the US, first findings in 2015. And just a note here, in Latin America, they sort of talk about maybe there's this other species that causes this fisheye symptom. We have found no evidence here in the US or Canada for that yet. We're certainly still doing some work to look for that, but at the moment, it all appears just to be Phyllocoromatus as the causal agent of this tar spot we're seeing here. Looking at a tar spot lesion up really close, this is the top side of the leaf across the top of the image here, and then the bottom side of the leaf here. So what it's doing when it's in the plant there is basically forming reproductive structures. So it forms these balls basically within the leaf and melanizes or produces this dark pigment, right, to help protect it from UV light, and then produces a whole bunch of spores within that tar spot. And so you can see some of these spores starting to form in here, and then they will get exuded out onto the leaf surface where they can be, moved onto another plant for continuing the infection process. The life cycle then of Phyllocoromatus, unfortunately, it can survive on infected debris. So there's been some work done here in the US to show that, yeah, unfortunately, it survives here in the Midwest. It has no issue with our freezing temperatures. That's not a problem. It survives. So initially, we'll see those black structures, those tar spot structures forming on the plants. Then it takes about 2 to 3 weeks to go through a cycle. So by the time a spore lands on a plant to the production of another tar spot, about 2 to 3 weeks, and then release of more spores. And because of this, because of only the 2 to 3 week time that's necessary and the number of disease spots that can occur on plants, it has a very strong exponential phase. And especially as we get into later in the season, the epidemic can really swing out of control very quickly. So what drives tar spot? I think the biggest message here is leaf moisture. So these are some sort of risk factors from some work that was done in Mexico, looking in the Latin America, like what conditions were driving disease in that part of the world. And these were some of the things that they found: sort of moderate temperatures, high relative humidity, leaf wetness, fog, and rainfall. They used some of those parameters to map it onto the US to try and predict where they thought tar spot might be a big issue. And so this map here on the left is just the predicted risk of where tar spot may be developing. It's relatively accurate, I think we'd agree, but we're seeing spread outside of this predicted range as well. And it probably doesn't account for some of the irrigated production, especially in Nebraska and Kansas. So there'll be some other factors to really think about. And I guess, unfortunately, we may very well see it move up into North Dakota too with time. Again, this picture just, I think, for Michigan really helps to drive home the significance of leaf moisture. So this is under irrigation. And unfortunately, this disease has really hurt a lot of our irrigated producers quite severely. In 2018, where we had a very wet summer, we had a very bad epidemic. Those people that irrigated saw, 50, 100 bushel losses potentially. 2019, you can obviously see where tar spot was driven by this center pivot, right, going across the field. But because it was a dry year and this is sandy ground, we did need to keep running that pivot. And from what I learned about this particular field, even though they drove tar spot under the irrigation, we still needed that, and that still out-yielded those dry corners. And just as a note too, that entire field had an application of fungicides. So we'll talk about fungicides here in a minute. Darcy, do you want to comment, I guess, about what you saw in Indiana this year?
Yeah, and I think you're sharing up there that shows the precipitation that occurred across the United States from June to August. And so if you look at that map, the dark blue areas is where we had a lot of rain. And in Indiana in general, the northern part of the state is where we've had issues from 2018 on. But as you can see, we've seen the disease move across the state, but as you can see, we have a band in the middle where it was dry. And so tar spot was there, but really didn't cause any significant yield impacts because we were so dry. But then in the southern part of the state, we do have pockets of wetness. And because the inoculum had already arrived in some of those areas, we do have severe disease that developed this season due to the amount of moisture that was available.
And Albert, we've left you off the map yet again. What can you tell us about your experience in Ontario?
Yeah, well, of course, Marty, thanks. If you want to flip over to the next slide there as well. And, unlike Michigan and Indiana and other parts of the Midwest, Ontario is a new player or a new partner or member of the tar spot group, as we always talk about. And first identified last year in late September 27th in the Ridgetown area, which is about an hour from Detroit. And subsequent surveys with the assistance of many of our CCA colleagues in Ontario, we're able to identify it in the lower 5 counties, but again, under low severity, low incidence. And we knew based on what Marty had said and the experiences in the upper Midwest that it would overwinter in Ontario, but to what degree? And we found that early in July, the same time as Michigan, Indiana, Iowa, Wisconsin, Illinois started seeing it. That first couple of weeks in July, we started seeing early infection of tar spot. What really surprised me, and I've been at this for 30 years, was just how quickly and rapid the development was in those fields that we had the disease in the past, but how widespread it ended up going. And you can see from this map that along the north shore of Lake Erie, right into basically the Toronto area, we had a wider than expected spread of tar spot this year. And again, that surprised me quite a bit, but maybe very similar to other experiences that we could see in new developing areas that are joining the tar spot party, I guess we could call it in some cases.
Absolutely. Maybe a party you really don't want an invite to.
Yes.
Right.
And just to re-emphasize again, we talk as pathologists, we talk about the disease triangle quite a lot. We preach that, right? So you've got to have all those factors together. You've got to have that susceptible host. The environment's got to be favorable that we talked about, but the pathogens also got to be present. So, for example, in Iowa, disease has been found all the way across the state, right? But yield losses haven't been that significant because it was dry in Iowa this year. So we really want people just to be paying attention to where this disease is. You've got the potential now in Iowa for potentially pretty significant losses if we get a wet summer next year or in years to come. All right, Darcy, would you like to talk about some of the work we're doing with hybrid resistance?
Yes, and we can we'll switch gears now, just talk about how do we manage this disease and what we're learning. So the first thing, is hybrids. Can we find hybrids that are resistant or what's the variability? And so in 2018, when the disease first hit us in Indiana, Illinois, Michigan, and Wisconsin, we went out and evaluated some of our hybrid trials, and across all those hybrids that we evaluated in all 4 states, every hybrid had some sort of susceptibility to tar spot, but the good news is there was a range of severity. So some of the hybrids did remain low, 1 to 5% of disease, whereas others, had 40 to 50% of disease. And so then we tried to take that data and say, how much, how is tar spot affecting our yields? And when we group everything together, we were seeing about a 1/3 to almost 1.5 bushel loss per 1% increase of that ear leaf being consumed by tar spot. But there is a difference in the maturity group. So the early maturing varieties, maybe it was only a 1/2 bushel loss per 1% increase of the disease in that upper canopy, whereas our later maturing varieties that provided that green leaf material later in the season as disease is ramping up, we did see about a 8/10 of a bushel loss as compared to 1/2 a bushel loss with every 1% increment of the disease occurring in the crop canopy. What this really tells us is it's really important to identify, that we may have hybrids that are tolerant, and this will help us manage this disease, but there is still variability. And even in our fields here in Indiana, I can go to a field that was significantly impacted by tar spot this season, and the next field over they planted a different hybrid, and its reaction is much different. So it is really important to understand there's variability in our hybrids, and really start asking if you're worried about tar spot, what those hybrids have and the relationship to how much disease can develop in the crop canopy. I think next slide, Marty, or is that? And so then the other question we went after is, how effective are our fungicides? And so we started back in 2019 looking at the fungicides that we currently use in corn production and looking at their efficacy against tar spot. And so we grouped these fungicides. We had 4 locations. So I had a site here in Indiana, and Marty had one in Michigan, and we had some in Illinois and Wisconsin. And then we evaluated them the last 2 years. So the last 2 years, I would say, you know, most of the states had less disease, so under a little less disease pressure, but variable pressure across the different sites. But when we're talking about using fungicides, I think it's really important to understand what they're designed for. So the fungicides are designed to be laid there when the disease is just beginning to start. They're going to provide us 2 to 3 weeks of protection. And the idea is we want to put them down where we're going to protect that yield. And so here, if I show this diagram, you know, in Indiana, in 2020, tar spot started later in the end of July. These trials were planted at the end of June, so they're a little later than some areas. So we go with our VT application about the 7th of August. We have a window of activity of 2 to 3 weeks. And then we start seeing tar spot ramp up. But you can see many of our programs will reduce that disease curve. So the non-treated curve there on the top, the blue line, you can see how we get that exponential increase in tar spot in the crop canopy. And when we put out our fungicides, you know, they're slowing that disease cycle down earlier in the season then, but once they start running out of activity, that's when we start seeing those curves ramp back up again. But the question is, you know, are we providing the protection at the right time to get us that yield protection that we need at the end? I think if you go to the next slide. So this is a group of the fungicides that we did evaluate in the last 2 years. And so this is new data that we will be putting out in a publication shortly. So all of these products were evaluated across the 4 locations in the 2 years. There's mixtures of different products, different fungicide resistant activities. So we have mixtures of strobilurins and DMIs, SDHIs as well. And we try to look at, you know, what products work, and then what are the combinations of products that we really need to control tar spot. Next slide. And so this is the summary of combining all that 8 years of data together. So it's across varying environments, which is good to show that we can see consistent disease control. So this is looking at the tar spot severity on the ear leaf at R5. And so the idea is how much of that ear leaf has been consumed by tar spot. So our non-treated control had about 5% disease on the ear leaf. And again, this is across varying locations. So I'd say Michigan and Wisconsin maybe had 3 to 5%, whereas Indiana, we were a little higher. But it's across all those locations. So we saw about a little more than 5% on that ear leaf. But many of our fungicides will significantly reduce it, keeping to 1 to 2% of disease. And so you see, we're starting to see a separation of products. Those labeled with an E are, you know, our best performing products. So there is a range of products available that we can utilize to try to manage tar spot. The other thing to note is when we look at the separation of the products, you know, one mode versus 2 or 3 modes, which we'll get into, does differentiate a little bit. And so then the next question is, yes, the products are working at on controlling tar spot, but you know, how do they impact our yields? And so for these trials in 2019 and 2020, generally there were 3 products that significantly outperformed the untreated control. And so you can see Veltima, Delaro, and Revitec were the ones that consistently across all 8 environments provided that yield boost. And I would say, you know, depending on the location and the severity of the disease, you know, the number of products that may have provided that yield boost will change, and it will change depending on how much tar spot develops in that crop canopy. And so again, the other thing we did look at is grouping the products together. So we looked at comparing, you know, does one mode of action work, or do we really need to go after products with 2 or 3 modes of action? And so by combining all those products together, what we're seeing is, yes, all the fungicides can reduce tar spot severity, but the 2 and 3 modes of action are really important in trying to manage the disease and get it as low as possible. And then the resulting yield on that, the 3 mode of action was the only one that significantly out-yielded the non-treated, but 2 modes is not that significantly different from the 3 modes of action. And this just brings us back around to, you know, when we're talking about using fungicides, we need to be cognizant of the risk of fungicide resistance. And so already, you know, we need to be incorporating more than 1 mode of action out there when trying to control this disease as well.
Mm-hmm.
And then the third thing we were starting to look at is, you know, what is the interaction between our hybrids and fungicides? And so I just pulled some data that we have in Indiana, and this is from 2020. And what we had done is we identified a couple hybrids, one that was really susceptible to tar spot and one that showed a little bit of resistance to the disease. And what we wanted to do is to see is would a fungicide be necessary if we can find fungicide resistance in that. And as you can see, looking at the severity of tar spot, our susceptible definitely has more disease. It had about 15% disease across the plots, whereas our moderately resistant line, kept it below 5. And that is very critical, I think, in moving forward when we try to manage this disease, that if we can find that resistance, that would be very beneficial. And as you can see, when we added a fungicide at the VTR1 application timing, we get the bump in reducing disease on our susceptible line, but maybe we may not need the fungicide if we can really find resistance, because there was no significant difference between just using the moderate resistant line and adding the fungicide on top of that. And then you can also see that in the yields. So again, our fungicide in a susceptible line significantly increased our yield in those trials, but there really was very little yield difference when we had a moderately resistant line available.
Albert, would you like to kick fungicide timing?
Yeah, so building on Darcy's comments there, particularly around the importance of the integrated tar spot management with hybrids, fungicides. Darcy mentioned the VTR1 application there and the response, and whether it's the tar spot working group or our individual trials in terms of fungicide trials for tar spot, and that timing is critically important in getting the most out of the products, getting the most out of the return on investment, and ultimately on yield and profitability as well. And so looking at the timings from non-treated through the various vegetative stages up to the tassel and reproductive stages, we consistently see, not only for tar spot, for many of the other foliar leaf diseases, that VTR1 application window has been the most consistent overall. But remember, we also have to be aware of the disease levels, disease development, when it's starting, and the local environment, but also the regional risk as well. And that's where the Tar Spotter app and that can assist us later on. But consistently, we see that the VT, R2, R1 applications are the best in terms of, or have been the most consistent over time. You can see double applications, particularly with an earlier application with that V6, VT application in this study in these studies across Wisconsin, Michigan, Indiana, and Illinois in 2019. But the benefit of that is still coming mostly from that VT application as opposed to those early applications. So again, getting the products where they need to be at the proper timing can lead to the best control and reduction in terms of both disease and maximize our yield there. And so having the products, having the tools available is critically important, but we also have to be able to assess our risk and determine whether we need it or not. And that's where the Tar Spotter app comes in. I've personally used this, and we've all used this. Our colleagues have used this quite a bit. I used it last year in 2020 to target where the highest risks would be for tar spot, to go out there and identify it, because we knew it was coming from Michigan, Indiana, and all that. And we also used it this year to highlight and promote and target our scouting early this year for that early disease development and follow it through into new areas. And Roger, do you want to take us through the Tar Spotter?
All right, I got to get my screen started here. Okay, so, and my phone started. Oops, sorry. I'm a kid when it comes to technology. All right, so this is a graph that my colleague Damon Smith at UW-Madison generated this season at one of his plots, and these are daily values that his tar spot model generates. And you can see it goes up and down during the season. And if you had this crystal ball at the beginning of this season, you would be able to know, oh, this is when I should maybe apply. And actually in this area, it was kind of had low probability. It was, you could have actually delayed maybe your applications here. So how do you get these values so you can see them? Well, you use a smartphone app. All right, so we actually have two smartphone apps. They have the same model in them. They attack it a little different way, and I can show both of those as you see them over on my phone today. This is actually what the Tar Spotter app would look like, and I'll start it up on the phone here. You basically use like 4 screens, maybe 3 screens. You set a location because the model works on weather, cloud-based weather. So you set a location, you can name it, and then you end up maybe with a list of locations. So they're on about a 4-kilometer grid, the weather. So maybe you need 1 or 2 or 3 for your farm, or maybe you have multiple farms or fields that you want to be watching. So you can have a list here. And this list right now has some different dates because I was using this during the season. And the actual meat of the model is this forecast screen. And you have a couple questions. The threshold right now is set at 40, but you can adjust that. You want to make sure that if you've sprayed fungicide, because that makes a difference. The growth stage, we're targeting V8 to R4. You can add irrigation if you had irrigation, and then you choose the date that you want to run it for. It will default to the current date, and it gives you a value. And you can also, during the season, send an email out that has a little CSV spreadsheet of the historic values for that area. So here I'm showing both apps. This is a different field that Damon Smith had calculated. This one had a higher risk. And what you can see is here when you're running the model on the 17th in the Tire Spotter app, you do see that it is high. And if you've been running it for the last few days, you're going to have a trend in your mind. The Field Profit app shows a 7-day trend for you, and that is this trend right there. Here's a little bit later in the season. On the Tire Spotter app, you'll see it's telling you 30 at that location. 30 is your percent. On the Field Profit app, here's the 30th, but you can actually do a little bit of forecast value, and now you have a chance to see that it's actually going to go high there. So what I'm going to do is I'm going to start up the Field Profit app actually and show you that the main screen there. So this is the field that has Damon's field there. I've already answered the 2 questions. We don't have any irrigation. And today's date, if I was running it, it's going to say, well, it's low because our temperatures are not right here. So I picked out California. I was looking for the right weather to make the app work. It's just where I wanted it. And on today, we have low going to high. And on this one, I can actually run the date a little bit forward, run the model. And now we see that in the future, a few days in the future, using forecast weather, it is predicting that you're going to have a high risk and you might need to take some action. So that is generally how the apps work. And I will turn it back to you guys. I know I went through it quickly, but and you can read the various points here on the screen.
Thanks, Roger. Sure, we're all set. All right, off we go again. Okay, so we've talked about a few things pretty briefly. We only have that hour. I'm sure we could spend the whole hour really talking about tar spot or fungicide efficacy. So what else are we doing? So we are screening hybrids and inbreds for their susceptibility or resistance to tar spot, working with corn breeders and our industry colleagues to try and assess that material. That's going to be very, very important for managing this disease in the future. We're also looking at the epidemiology of this disease to better understand exactly what's going on in terms of spore release, when is that occurring, and what is the impact of residue and tillage strategies for this disease, some genomics and host pathogen interactions, and some other sort of detailed work we're trying to do to better manage this disease. We've certainly focused on tar spot here, and hopefully, if you haven't dealt with this in the past, we just really want you to understand that it can be a very, very damaging disease. But at the same time, we certainly don't want to forget about our other diseases that are out there, right?
Oh, absolutely, Marty. And when it comes to some of these, it depends on where you're at and what the history has been for many of these foliar leaf diseases. And many of them are endemic throughout the US and into Ontario. And that good example is northern corn leaf blight. This year, we've had resurgence of northern corn leaf blight in many locations here and in other areas. And in our trials, in our inoculated trials, we were seeing 60 bushels or more to northern corn leaf blight. So again, very important, economically important disease. We've seen the impact of particularly, again, these regional high-risk pathogens or diseases such as gibberella ear rot. We've seen this year in certain parts of New York or other states where gibberella ear rot and dawn issues. 2018 through Ontario, where we had considerable impact there. So a lot of these diseases, tar spot, is an important disease to consider, but don't forget about those other endemic diseases that can consistently cause yield issues to not only yourself but your customers as well, have to still be considered in there when you're planning for your 2022 management down the road as well. And this is where the Crop Protection Network comes in as well to assist with that, with many of the different tools out there as well. And that, right, Marty, as we go over to the next slide there, and talk about the fungicide efficacy tables there, disease loss estimates. We have many different information materials and products available for these different diseases to assist you in terms of managing these and dealing with them as well.
Right, and just to point out, we do have task spot on that fungicide efficacy chart. This is last year's. We'll be updating that this coming February, so there'll be some improvements to that, some revision of that, given the additional data that we've collected this year and in the past couple. So keep an eye
and many of the information out there and on the cropprotectionnetwork.org website also has the availability for CCA credits as well. So there's another great opportunity besides the 8-part series here sponsored by the CPN, but also go to the cropprotectionnetwork.org website and you can build on some of those CCA credits as well as learn about these diseases.
Absolutely. And in terms of managing task spot, then, I think one of the most important pieces of this is going to be that hybrid resistance or tolerance. Like as Darcy said already, there's no hybrid that's completely immune to this disease, but there's definitely differences between them. So trying to identify those is going to be very, very important. And of course, weather, is the key driver here. If we have a dry season again, it's not going to be as big an impact as it was in 2018 and 2021. We talked about Task Spotter just now, but certainly scouting is very, very important. That Task Spotter app is fantastic for trying to look at risk, but we need to know where the disease is at. So again, if you're in an area that hasn't had Task Spot yet, you need to be out there scouting. And then in terms of, in season, like what can we do? We certainly, if we have Task Spot developing, we want to look and check for lodging potential. So certainly this disease can take yield, affect test weight, but the other really big impact here is that potential for lodging. So we can have some great corn, but it can end up lodging because of Task Spot. So being very mindful of that. We're continuing work in terms of crop rotation and residue management, but we've had plenty of instances, where we've had fields rotated or a field that hasn't been in corn for many years, planted to corn and then get basically smashed with Task Spot. So the problem is it's a regional issue. If there's fields around you that are infected, these spores can travel many miles and inoculum can come in. So even if you've never had corn in that particular field, or you're doing all the right things in terms of crop rotation, it's not going to completely eliminate disease. Of course, if you're a high residue and you're no-till corn on corn, the risk does increase slightly. But again, it really comes back to that weather and how that's going to drive disease for coming seasons. And we'd like to thank all of the sponsors that make this research possible in our states that we're representing and others. Without that, it just wouldn't be possible to bring that. And then Albert.
Yeah, in terms of the Crop Protection Network, although Marty, Darcy, myself, and Roger are here representing our institutions, we have a vast network across. It's an international network across the U.S. and including Ontario here, and you can see the many different land-grant universities that are participating in this. And again, that cropprotectionnetwork.org is an excellent resource for not only tar spot but other diseases, and also that opportunity for those increased CCA credits as well.
All right, and once this webinar concludes, there will be a poll that we're putting up there. You have to be 18 or over to participate, and it's at your free will, but we would really appreciate you participating in that. What we're trying to do with that is get some sense of the amount of yield loss and put some economic numbers to that, and certainly changes in fungicide practices. It's pretty quick. I think there's 6 questions in total. So if you could do that, that would be much appreciated. And this is our final slide, and we'd very much like to take some questions around this. What can you do? Well, we've talked about some tools that are out there, right? Assessing risk is very, very important. Is it endemic in your area? And if so, scout, or if it's not endemic to your area, still be out there and scouting. And this IPM pipe corn website is where we put up these maps that are essentially real time. Particularly if it's in a new state, we try and log that so we know where the issues are arising. Essentially here in Michigan, we really don't need to do this again because it's across pretty much all of our corn producing area. The risk is there. So we don't really need to push that map in Michigan, but certainly into the fringes of where it's developing next season, that's going to be important to keep an eye on things. Talk to your seed salespeople about hybrid resistance. We talked about those differences. If you make a fungicide application, they can certainly help, and I think we've all sort of witnessed those, protection of up to 50 bushels or so, but make sure you leave some check strips. I think that's really important to understand what actually happened. If you don't have the checks, it's sort of hard to ask that question. And if you're not sure about whether you've got the disease or not, submit samples to your diagnostic clinic. As we just touched on, let's not forget about other diseases. So any final comments from Darcy, Albert, or Roger?
No, we can take questions though.
Yes.
Absolutely.
Great. Well, I do have a nice list of questions coming in here. They're all quite varied, so there's no reason but to go straight down the list, and these would coincide, obviously, with the time that they came in. I think most of them could be addressed to any of you, mainly Albert, Darcy, and Martin. Roger, I do have one that's pretty app specific, so I'll ask you that one specifically as we go through this. But here's the first one. Is there any evidence of tar spot being seed-borne? This person says the first initial field location found in Pennsylvania, as well as the first in a new county in Pennsylvania, were in a variety hybrid trial. Perhaps it was found more readily because of more people looking in those fields, but it's a very curious coincidence. Sounds like Darcy's our responder.
Well, I'm going to be responding. Yep. So from the literature, and as far as we know, right now, tar spot's not infecting the seeds. The seeds aren't the infection point, but the question is, are we moving it on the debris, right? And so how small of that debris does it have to be moving around in our corn? And so that's a possibility of one of the questions of how is it getting distributed? Is it can be moved around in the debris if it's the seed, if it's contaminated, the seeds are contaminated with that debris. We're still trying to explore that. How far does the infection go? And so that's some of the questions we hope to answer in the near future, but it takes time.
Yeah, I think if it was truly like seed-borne, I think the disease would have been here much earlier, given the amount of breeding we do across, northern and southern hemisphere.
Okay, thank you. Moving on to the next question, a much shorter one. Corn silage. Is there an impact of tar spot on crude protein with corn silage?
Crude protein. I can't think to the crude protein element, but certainly it affects quality of silage without a doubt. So I did a sort of quick and dirty look at this back in 2018 when we had that bad epidemic, and there was huge differences between areas that were getting, they look like they're frosted off, right? Areas of the field that were just dying compared to another area that was still green. I think first and foremost, it dries silage down so quickly, so you've got to be very mindful of that. Do we have the correct moisture content for ensilement? But yes, it absolutely affects quality parameters, basically leaving behind a lot of non-digestible components, right? So it's digesting those good carbohydrates you want to feed to your cattle. The protein, Pete, I'm not too sure, and I'd have to check our numbers.
Okay, thanks, Martin. All right, moving on to the next question, getting into irrigation. Do lawn drop tubes reduce disease under irrigation?
I'd say yes. No one wants to do it though. So if you want to do that, we would very much like to partner with you to outfit a section. I I have no doubt that, so it's been an issue when we've had here in Michigan talking to, and I guess a little bit in northern Indiana, right, where we share some of that seed production ground. Where we have run irrigation, it dramatically increases the amount of tar spot. There's no question about that. That's all overhead. If we could run drop tubes to remove moisture from the canopy, I would bet money that that would help. But I don't think anyone wants to deal with breaking drop tubes and the hassle.
Okay. All right, moving on to the next one. This is about developing a resistance for maintaining resistance in our genetics. And that is, would the use of fungicides paired with resistant varieties be the way to go to maintain genetic resistance in our crop plants?
Albert looks like he's got the answer.
Yeah, so basically in regards to that, as Darcy had mentioned, from our integrated management practices, we're seeing both using highly resistant or, our best tolerant hybrids out there with fungicides is our best attack on this disease. In terms of, we don't know enough about the resistant side right now to know what's providing us with that tolerance or so in it. So I'm not sure how to answer that one, particularly in regards to that. I don't know, Marty, Darcy, what do you think in that?
I agree, it makes sense, yes, absolutely. Like reducing pathogen pressure, but I think it's going to be a difficult disease to breed for. There are some QTLs out there at the moment, but they contribute, only a certain amount of resistance to tar spot. That's why we need to be partnering with industry and doing more screening of inbreds. And it's going to take time as well to bring these brand new hybrids through. It's somewhere between 5 and 8 years, depending on the method, to bring new hybrids through to market.
I would say we're also, we also see differences in how the fungicides are reacting on our different hybrids too, as well as managing it. So we're starting to be able to tease some of that out, but it's just taking time to get to that point.
Okay. So a lot of it comes down to spore load too, right? And I was, as I said this year, I was incredibly surprised with how quickly the disease developed and how widespread it was. And I wouldn't have thought we had a lot of, the load wasn't there to develop and build up.
All right. Well, very good. The next two questions that I'm going to go through are associated with the app. They came in at that time when Roger was giving his demo. First one, I don't know who wants to take this one, but what does the action threshold actually mean? Can we give a little bit more of a description about that term?
All right, so the action threshold, which we default to 40%, is where we've determined is a good point where you should actually think about applying a fungicide. And we know that some fields maybe have a higher history of that disease, or some fields maybe have a greater resistance to it. So we allow them to change that action threshold a little bit down or a little bit up. It's, in a way, it's arbitrary. It's like an alarm clock. It sets it at 40 and it turns it red. And so you really know you got to take some action. And 40 has been the one in the last 2 or 3 seasons when we've been running the app, that if you can do something and when it hits that level, you have a good chance of protecting your crop in the next couple of weeks with that fungicide application.
Okay, thank you, Roger. Next question here, again, associated with the app. This person says, it appears that the Tar Spotter app is saying the risks are highest a number of weeks prior to tassel, but the timing study indicates that VT is the best time for fungicide application. How do you weigh those conflicting pieces of information?
Okay, as someone who actually knows all the numbers that go into the app and how the weather affects it, early in the season, what's happening is the temperatures are generally cooler just because of the time of the season. And cool temperatures are affecting the model and causing that percent risk to be high. So it doesn't always happen that way, but if it does, you still have to kind of wait until until it's in your crop is in a vulnerable state. So in the Arlington field, it was high coming right into the the R8 to VT reproductive state. And so they actually sprayed at V10 in that situation because they could see that it was high. And so the model responds to weather. The weather may be cooler earlier in the season. It is a little bit of your own experience too on whether you're going to pull the plug, or pull the trigger and spray that early in the season. But the model is just watching the weather. And if those cooler temperatures come later in the season or come earlier in the season, it's going to let you know that that weather is conducive for that infection.
Now, Roger, the growth stages for tar spotter and that is that V8 to R4, right? That's
right.
The key for that side as well. So again, as Roger's mentioned, it's a tool to aid in assessing your risk, whether to spray, not to spray, and that. So, as with any of these forecast prediction models, it's important to be out there too and assessing and determining it. Just, the app may show or may not show, but nothing is better for you or your customers to be out there and being aware of the disease.
The app is trying to make sure you're not surprised. Sometimes you don't understand the weather as much. It was hot the last week or the last week and a half, and you forget that there was some cool weather in there. The app doesn't forget. It doesn't care. It has a 2-week memory basically, and it doesn't care if today was hot, if there was just enough coolness and moisture in the past. It has found that infection can be triggered.
And I guess I could comment on the changes that we've seen here in Indiana. And so I would say this year, we shared the data where we're consistently showing that between the VT and R2, the fungicides laid there seem to be working best. And even here in Indiana, when I look at this timing that we've done this last season, that window still works for us. But with the app, it triggered us to go a little earlier. And so we do have those treatments that went out a little earlier, and then they triggered a second application. So that, depending on your field location, will dictate what your risk is. And so those are fields that had high risk of tar spot. Not the whole state of here in Indiana had that high risk, but those fields at high risk, the disease did kick up early. If we had gone out that V10, V12 application and then hit it again 3 to 4 weeks later, when the app triggered again, those were our best performing plots. But when we look at all the combined data, still going out at our standard BTR1, R2 timing, still held pretty good this season. We didn't share all that data. We're still compiling it, but I think the app will help you hone in, especially if you have a high risk situation, and it'll give you an idea when to start looking for the disease. But I don't think we need to significantly shift our management practices to try to control it. Just understand these are tools that can help us make those decisions and make an informed decision to put the fungicides out when needed.
Yeah, and absolutely, and also important to consider those other diseases, other risks, whether it's your field or regional. And if you look at the Great Lakes and what we've seen, not only this year but other years, as I mentioned with gibberella ear rot and Dawn, that R1, that silking application is critical to managing those gibberella ear rot and Dawn mycotoxin production, particularly in those high risk fields or high risk areas as well. And if we go early and late, we'll miss that window as well.
All right. Looks like no more comments on that particular issue or question. So I'm going to keep moving along. We had this question kind of repeated in various ways. So the way that this individual asks it, I think is best. So this person says, I continue to hear speculation that the fungus can produce a toxin which is responsible for fisheye lesions, poor standability, and other issues. Is there any evidence of this whatsoever?
We don't have any at the moment, no. We don't really know why we see those fisheye symptoms. We've done some sort of DNA work to look at who the microbes are that are in those fisheye, and we're continuing to do some of that. We don't have a strong evidence of 2 different strains, one causing fisheye and one not. But yeah, I mean it's a good question, but we don't know the answer.
Okay, thanks, Martin. Going on to a different type of question here. This person says, how receptive have seed corn companies been towards being involved in research and helping producers understand which hybrids provide moderate resistance to tar spot? This person says it's been a constant question producers bring up at seed meetings. So he's wondering if seed corn company involvement has begun to increase as tar spot becomes more and more of an issue. Any comments on that one?
I mean, I can speak to our experience here. Yes, there's interest, and we are trying to engage more with them, I guess. I mean, we have, us as a group, have a corn disease working group where we try and, interact with industry every year to make sure we're, communicating, right? Locally, yes, seed companies have been working with us, not all of them, but a number of them have been looking at their hybrids that are sold here in Michigan to see how they perform. And I think that's valuable. So that's been my experience.
It's been similar experience for us. Again, it's new for us, but we had a hybrid trial next to our fungicide trials, and we had constant communications and visitations to those trials from the companies as well. And also in our corn performance trials, we were able to utilize those this past year. And the three locations in southwestern Ontario that had a lot of tar spot, we were able to get those rated in that, and we just presented that information to the seed corn companies in Ontario just this past week with Dave Hooker and I, and there's a lot of discussion between us and the commercial and public breeding programs in trying to figure out what's going on from the resistance side. And again, it goes back to some of the previous questions. We don't know what's going on in terms of resistance, partial resistance. Will that have that impact on inoculum or disease development, and then subsequent spore production and the longevity of these tolerance aspects of the hybrids there? We don't know this at this point. There's still a lot that we don't know about this disease, or this pathogen.
Sure. Thanks, Albert. Okay, I had a lot of questions come in around management of this. The three big ones that I'm seeing, one, and you mentioned some of these. Does crop rotation help mitigate the risk? Does tillage also do so and potentially break down residue and help in some way? And three, is there an impact from early maturing hybrids, sort of avoiding some of the issues? Any comments on those three things? Again, crop rotation, tillage, early maturing hybrids.
Well, I guess I can go. So we really don't see differences in crop rotation and tillage in these areas that we have such high inoculum loads. I mean, the history of the field will dictate that the disease may start in that epicenter of a field that's really bad, but once those spores are moving in the regional areas that have the tar spot, it really is going to move to field, and really rotation's not going to have that impact. And we've seen the various fields with different management tactics, rotation and tillage, still being highly impacted. So we're still trying to figure out if there are ways that we can minimize that, maybe with some tillage or rotation. But again, we're really not seeing that be a significant factor in trying to manage the disease. And then on the third, and I forgot what the third question was, on
early maturing hybrids.
Early maturing varieties. So you could think about that. I guess we were having this conversation earlier with Albert and Marty on, should we switch to early maturing varieties in areas where the disease is worse? And I guess right now we're not at the point of saying to change any of your management practices. Still select those hybrids that are best for your region, but understand that, depending on the season, and on those environmental conditions, it's the amount of green tissue that's available. This pathogen is going to go after the green tissue when it's really ramping up. So maybe, and those later maturing hybrids have more green tissue at the end of the season, so that's why they're showing more disease for this year versus another year. But again, we're not suggesting switching that, but just understanding more. Is there hybrids that are more tolerant, and using an assortment of hybrids on your farm, not just relying on one, would be highly critical as well.
Okay, thank you, Darcy. I think we have time for only one more question. And this is a good one. This person says, is it rainfall that drives tar spot and the level of infection, or is it a lot to do with leaf wetness? This person says they had a severe drought in their area, but because they had foggy days and some moisture buildup on the leaves, they still saw tar spot. Any thoughts on that?
it's moisture either any way you get it is my thought on it, you know, whether it's long dews or you're near a river bottom. And so that's where we have bigger impacts in Indiana, is along our river bottoms down in southern Indiana. But rain is also going to contribute. So it all depends on what's going on in your environment. That these long morning dews, when we saw that this past July, those are things that are contributing to the disease to develop.
All right. Thanks, Darcy.
And I
Any other
think
comments? Yeah.
And I think when it comes to that whole leaf wetness, whether it's humidity, rainfall, etc., I think that is the key here. I know, Roger, with temperature and etc. here, but that leaf wetness, that moisture drives a lot of this disease. And that, based on our experience, we had temperatures that were exceptionally high this year, and the disease continued to develop where I would have expected it to slow down. So those cooler nighttime temperatures and leaf wetness can really drive this disease to the point that we can end up with epidemic conditions in some cases as well.
Okay, thanks, Albert. Any final comments, Martin, Darcy, Albert, Roger, before we wrap up today? Nothing? Okay. Well, I do have a lot of questions left. And so, folks, what I would suggest is, if you want your specific question answered, I try to get through as many as I could and kind of get the representative ones answered. Please reach out to them individually. I'm sure an internet search alone will yield good results for finding their phone numbers and emails, especially since university connection here. So with that, thanks to each of you, Martin, Darcy, Albert, Roger, I appreciate you being on today. Thanks again to the Crop Protection Network for sponsoring today's webinar and the larger series. So be on the lookout for coming webinars. And also, as was mentioned during the event, look at the Crop Protection Network website. A lot of other resources there for you to learn from. So with that, thank you everyone for joining today. We look forward to seeing you with webinar 2 in this larger series. Take care, everyone. Bye-bye.
Thank you.
Thank you.
Bye-bye.